after a.m. Can you hear not so good, but I think it's my time.
It's your time, yes.
Okay.
So it's a great pleasure to introduce you to Professor Cristina Varese, a full professor at University of Turin, the scientific head of the Turin University Culture Collection, and the coordinator of the national node of the Microbial Research Infrastructure MIRRI.it, that you will soon hear about. And we are very happy and honored to have this keynote lecture, also because I know, Cristina, that you will soon fly to Portugal to represent Italy in MIRRI network. So welcome to our training.
Thank you so much, Laura. Can you hear me quite well?
Yeah, super well.
Okay. So thank you to Laura, thank you to all the organizer to give me the possibility to explain you a bit more about this European research infrastructure. And I will try. Okay. What does it mean, MIRRI? MIRRI means Microbial Research Infrastructure, is the EU research infrastructure dedicated to microorganisms, to find solution and to provide solution to both academy and bioindustry, and is a pan-European distributed research infrastructure. And the main aims of this research infrastructure are the preservation, the characterization, the distribution and valorization of any kind of microbial resources. Here, I hope you can see my pointer. Here you can see the website where you will find all the information I will provide you today. So first of all, why microbes? Because microbes are really, first of all, are the most abundant form of life in our planets. So we can't see them by naked eyes, but they are everywhere, are really pivotal for any ecosystem services in both terrestrial and aquatic system. And right now we are already aware that microbial diversity is really important for science and industry. Here you can see some of the already well-established microbial valorization in different fields of application, like waste valorization, bioremediation, primary production, and of course, food and pharma application. And so now we are also aware that microbial diversity has economical and financial value. And we have to keep in mind that right now, more or less 90% of the microbial diversity is still to be investigated and characterized and preserved. So there is really much of work for all the person working on these fields. And indeed, now we are facing many serious societal challenges, and we always think about bioeconomy and sustainable economy and so on. In my opinion, microorganisms are really the engine of the bioeconomy and environmental sustainability. Through microbial valorization, we can really promote global development and human well-being. And I hope you can see here the different ecosystem services that microbes can provide to us, which in turn are equivalent to different deliverables that can help us in achieving different sustainable development goals. Laura, sorry, can I tell you if you see this part on the left of my screen or not, or for you is okay?
Yes, we can perfectly, and we can hear you very well.
Okay, thank you so much.
Thank you, Cristina.
So MIRRI, when we start working on MIRRI in 2012, the European situation was a fragmented situation in which different cultural collections were already present. We were aware of each other, but there were no organization in terms of data, in terms of services, in terms of what we can really supply all together to the lay society. And so from this picture, we try to get this kind of picture in which we try to harmonize everything. We try to create a European catalog of microbial resource. We agree on the minimum standard for the data set in order to allow a verification of our catalog. We try to put together the expertise in training course, education, and in consultants to private company, and so to work really as a European research infrastructure. And first of all, the most important things is that in the website, or I already show you, you can find everything you really need in terms of microbial resources. And this is also one of the reasons why we are working according to a FAIR principle. Everything is open access and everything is available for anyone. So simply scrolling in the website, you can find all the data about available data, public available data about the more or less 200,000 microbial strains that right now are present in the MIRRI catalog, but you can also get many other information. So when you consult our website, you can find, for example, the kind of services we can provide for both academia and bioindustry, starting from isolation to preservation. And I just want to give you one of the reasons why we started to really highlight the importance of preservation. In a survey in 2008, we discovered that less than 1% of the described microbial biodiversities in important papers were not available anymore in any European collection. That means that EU, in a broad sense, spend a lot of money in isolating and characterizing microbes for important ecological or biotechnological investigation. And then the strains simply disappear because have been contaminated, or the professor in charge of the research collection died or retired, and so on. And this has been quantified as a huge waste of money from the EU community that state that it was not possible to continue in this way. So we start really to highlight the importance of the preservation of what we call key strains. Key strains is any microbial strain which is a new species, of course, or is representative of important ecological services provided by one strains, or is a strain that is important for a biotechnological point of view, or is a strain which is important because, for example, is a pathogen, or so on. So in any of these cases, we kindly support the idea that this strain should be deposited in a public collection and should be available to all the scientific community. Then other important goal of MIRRI is the taxonomic characterization of the strain, the data annotation, the systematic investigation, the distribution the fair, let me say, the fair distribution of strains, because again, almost at least in the past, the availability of the strain was more related to the friendship with the owner of the strain, with instead of an open request of strains. And this is really not polite. So distribution should follow fair criteria. So any request have to be transparent and the delivery should be transparent. And last but not least, MIRRI also provide many training and service development. So here, just to remind you the long way we had. So we started, as I told you, in 2022, when MIRRI entered in the S3 roadmap. And then phase by phase, we became an ERIC, so a legal entity recognized by the European Union. And any day there is the possibility for new countries to enter in this system and to support the microbial biodiversity valorization. So right now we have 6 countries, the one you can see in green, which are member or observer of the MIRRI-ERIC, and 2 other countries which are prospective members. So in the negotiation of entering in this distribute pan-European system. And here in the left, you can find the administrative organization of MIRRI-ERIC. So there is an assembly of members where the representatives of each country are seated, and they are the decision-making arm of MIRRI, and they are assisted by an ethical board and by an advisory board. The assembly of members tell to the executive director and to the central coordinator unit what they have to do for the different activities. And in practical, any national node then will take the activity according to what legislated by the Assembly of Members. And any national node is a coordinator. I'm really proud to be the coordinator of the Italian national node. And we, national coordinator, meets regularly in a national coordinator forum. MIRRI want really to be a tool for foster microbial biodiversity valorization, and hence you will find the catalog of the microbial resources with all the associated data provided by any national node. You will find you will find all the strains, including the reference type for your research, but you will also find the list of services and all the other possibility to interact with us. I will explain step by step all the opportunities. So as regard the catalog, I already told you the catalog of MIRRI more or less encompass 200,000 high quality microbial resources. I want to highlight high quality because the microbial biobank are the institution where the taxonomy and the systematic of the different microbes is really important. So we take care of the continuous updated of the nomenclature. We recheck from time to time all our strain to be sure about the identification. And of course, together with the microbes, we also provide information, which are the so-called metadata about the taxonomy, ecology, pathogenicity, morphology, physiology, chemical characterization, and whatever you know, genomes and so on. So any information which is public is added to the catalog. And to facilitate the search on the catalog, you can also search according to your area of interest. We highlighted 6 different areas, one related to human and animal infection disease, another related to biopharmaceutical and therapeutic solution. Another is more related to food and feed production. The area 4 is related to soil and crops. Area 5 is more related to biomonitoring and bioremediation for different soil, water, and towards different kind of pollutants. And last but not least, area 6 is related to bio-based chemical, new materials, and bioenergy solution. So you can search immediately if you are searching a precise species or strain in the general catalog, but you can also search according to these criteria. And here is just an example of the screen you can get in our website. And so here are the different, as regards filamentous fungi, you can find 6 different groups of fungi according to the different area. And for any of these fungi, you can again search in the strain catalog or find a taxonomic catalog. That means, for example, that if you are searching with an old name, we will immediately tell to you, you are searching with an old name, the new name of the species is this one, please take it into consideration. And then after the catalog, we provide many other kind of services, and these kind of services are tailor-made for different kind of stakeholder that could be researcher in a different institution, but also bioindustry and policymaker. And so again, there are 2 different kind of search tool. One that is a general service catalog, and one which is already tailor-made for industrial application. And so here is simply a short list of some important general services that the different microbial biobank can provide to you. Supply of microbial resources. Probably I forgot to tell you that in the catalog and in the resources of MIRRI, you can find any kind of microbes, so viruses, archaea, bacteria, cyanobacteria, filamentous fungi, yeast, and microalgae, plus other kind of materials as cell line, plasmids, and DNA samples. So supply of microbial resources, public, patent, or safe deposit. Sorry, I made a mistake here. The identification of microbial isolate, the isolation, preservation, and cultivation, the long-term preservation. What happens?
Nothing. Apparently nothing.
We can hear you.
You can go on, Cristina. No. It's okay, we can hear you.
Just show your screen.
Cristina, can you hear us? Can you hear us?
Can you see? Can you hear me, Laura?
Okay. Everything's fine now.
Okay. So, for example, several private companies ask us to freeze dry for a long-term preservation of some of their specific strain, or to set up an optimization protocol for their preservation. We are also working for the in optimizing protocol, tailor-made protocol for the cultivation and fermentation, both in submerged fermentation and in solid state fermentation of different kind of microbes. And then molecular typing, microbial counting, screening, karyotyping, plasmid profile analysis. So you can read by yourself. The list of general services is quite long. And as regard the application of specific services, again, I kindly suggest you to have a look to the website. I here only reported some of the topic on which MIRRI has already developed some specific biotechnological services for all these sectors. And Laura, please feel free to ask me to stop if you want any further clarification in what I'm telling you. And
Cristina, one moment, wait. We asked to the public if they have questions. So, do you have anything to ask to Cristina up to now?
Yes, I want to ask.
Yes, there is a question.
Sorry, I didn't catch where physically the organism was. In which laboratory?
Cristina, can you hear? The question?
I think I catch the question, and the answer is right now MIRRI encompasses more or less 50 different microbial biobank, which are located in the 8 countries I already show you. So if you need any microbes, you can ask through the website, and the secretary immediately requests to all the collection that is preserving the strain, who is available to provide you the strain.
Okay. So the answer was 50 countries are about 50 countries.
Different.
Yes, and there is a list on the web page. You can consult the list and ask directly to the owner of the culture. Am I right?
Yeah, you can ask to the owner of the culture or through the website, and we will search for you.
Okay.
For example, Cristina, do you know if Roscoff culture is in the network?
Roscoff is not in the network right now because Roscoff is belonging to another research infrastructure which is called EMBRC, and is specifically dedicated to marine bioresources.
But through our
my culture there.
Through our project that we have with Cristina, that Cristina is leading, even cultures from CNR will get in the Italian Microbial Culture Collection Network. And if we achieve standard quality, then we can become part of this network.
Yeah, sure.
Okay, thank you. We can go on.
And Laura, I only focus on MIRRI, but perhaps it's better to remind also that there is another important organization in Europe, which is called European Culture Collection Organization, which is...
Maria will talk about this on Wednesday, tomorrow morning or Wednesday morning. So Maria will talk about MIRRI and the WFCT. So we are covered.
USCT?
WFCT, World Federation of Culture Collection.
No, I was mentioning ECO, European Culture Collection Organization.
Maria will have a lecture mainly on ECO.
Okay.
So she will present the network.
Okay, so wonderful. Coming back to MIRRI, another kind of services we provide to all the different stakeholders is the expert clusters. Expert cluster means that there are a group of persons which belong both to the microbial biobank or also to external organization which discuss together and try to find solutions which are posed by policymaker or by industry. And right now there are 5 expert clusters. So ones related to legal, regulatory issue and standards. Just to give you an example, Nagoya Protocol implication for microbial biobank, for exploitation of microbial biodiversity, for digital sequence information, and so on. Application in biotechnology, taxonomy. Sometimes we like to discuss a lot about the taxonomic position of specific microorganism. Setup of new protocol for bioprospection, cultivation, and preservation. This is really important for us in terms of quality management system of a collection. And high technology as, for example, MALDI. MALDI-TOF is becoming the gold standard for microbial identification since it's really cheaper and faster respect to other molecular techniques. And so indeed, I will be in Portugal this night because it will start a huge project in which we will try to create an open access multi-spectra database of about 150,000 spectra to allow the fast identification of the most important microbes in any sector very easily. So that's just to give you an idea of a project we are trying to perform. And of course, we also have internal working group which try to face the most important challenges in terms of technique. So, for example, whole genome sequencing or MALDI-TOF. But we are also trying to make a step further, not only in the preservation of single strain, but also in the preservation of microbiota. So, for example, in the Italian project I will show you in a few minutes, we had a group working on different kinds of microbiome, plant microbiome, soil microbiome, water microbiome, food microbiome. Any of these microbiome has specific needs in terms of understanding which is really the microbial diversity present in this kind of community and how we can really preserve the community in terms of taxonomic biodiversity, but also in terms of functional biodiversity, since if we want to reutilize microbiome, we have to be able to preserve this microbiome. And this is something we are still working on. And finally, in the website, you can find a lot of possibility of training and education, again, for different kind of stakeholder. Here you can see the website that immediately give you the possibility to scroll the training. And I just want to underline that you can select your training according to the language, according to the possibility to follow the training course on site, online or in a hybrid system, according to the duration, to the country, to the organist group, to the application domain, and so on. So again, since there is more than 100 training courses you can apply for, we can try to make easier the selection of the training course in which you are interested. So what I hope I was able to describe you that MIRRI is really a working collaborative environment in which we hope to provide solution for researcher, for industry, and also for policymaker, to really foster the research and the transfer technology in different biotechnological sectors. Right now, we are involved in about 10 important European projects that are the one you can see here. And we have a lot of collaboration with many other research infrastructure. The one that I cited about Roscoff is this one, EMBRC. And as I told you, is focus on marine biotechnology. And what now I want to tell you is there is a great opportunity to take advantages of transnational access. What transnational access means? Transnational access are program we've found you for giving you logistical support to get access and to use the facilities of the different research infrastructure, in this case, the one of MIRRI. Right now, there is an open call that will end in the end of June. Here, if you are interested in taking advantages of this possibility, is the link to apply. What does it mean to apply to a transnational access? You have to write a short project in which to underline why it's important for you to get for free the services that are offered during this call. And there is a panel that will select the best project, and the person that will win the call will have free access to the services and some money for supporting their physical access to the different institutions. And now a few words on the Italian network of the microbial culture collection. First of all, I want to tell you that right now there are 27 different institutions in Italy which belong to the JRU MIRRI, and we are really very proud of this joint research unit. And Italy is, how can I say, it's really a strong country because it's one of the few in Europe that host cultural collection working on virus, bacteria, yeast, filamentous fungi, and microalgae in the same country. We started to work together in 2017, and we had one of our goals was to improve the quality management system in the cultural collection, but also to try to understand which were the needs of the stakeholder to be able to support them. In 2022, we were really very lucky because we won a huge project which is called PNRR SUSMIRI, with a total budget of about 17 million euros to strengthen our facility with cutting-edge technology, to implement the services we can offer to both academia and bioindustry, and also to address the weakness of our network. So the creation of the Italian national node really gave us the possibility to harmonize the service offered by the different cultural collection, trying to avoid redundancy and to increase the complementarity, and also to address some issue related, for example, to the compliance of Nagoya property, to biosecurity and biosafety regulation, intellectual property rights, and so on. And we are also working on overcoming fragmentation and trying to maximize the effort in identification and preservation. And we use this money also to buy cutting-edge technology in a complementary way, in the sense that each of the different institutions focus on different aspects. And the idea is that all the cutting-edge technologies we have acquired through this project are open access. That means that any of the other institution can get access to this technology without any problems and in a friendly way. We are trying to enlarge our national network, and we are also set up again a series of training courses, avoiding the overlapping between the different services and trying to merge together services provided by different institutions. So here you can see very, very quickly the 6 work packages which has been carried out during this project. I just want to focus a little bit on defining common quality management standard procedure. It's really very important. During this project, we were able to write down 12 procedures that each Italian cultural collection has to certify in terms of acquisition, characterization, preservation, and distribution of microbial material. Of course, these procedures have to be adapted to the different group of organisms, and so there has been really a huge work by all the partners to try to standardize as much as possible these procedures. And another thing important is the disaster plan. Each institute has to provide a disaster plan in which try to highlight how, in case of earthquake, flooding, or whatever you can imagine, they can save in a master collection deposited elsewhere their collection. As regard the work package 3, we are really very proud because we were able to create for the first time the Italian microbial catalog. So right now any person in Italy can immediately understand where a specific strain is preserved and can be immediately put in connection with this cultural collection. We also supported each cultural collection with a software for the manager to increase the database management. We create the Italian catalog in order to be interoperable with the MIRRI ERIC one. And finally, we are finishing to develop some important bioinformatic workflow. The 2 most important bioinformatic workflow are related to the whole genome sequencing. One workflow is designed for the automatic annotation, assembly and annotation of genome of viruses, bacteria, and fungi. And the other one is related to the analysis of microbiome. When we talk about microbiome, in most of cases, colleagues are referring exclusively to bacterial community. This is not fair, is not polite, but when you ask why you are only taking into consideration bacteria, in general, the colleague responds, because I'm not keen on fungi or viruses, and I don't know how to manage this data. So we try to help the scientific community making available a workflow that immediately gave you the possibility to analyze bacterial, fungal, and viral community all together. As regard microbiome, I already told you that there are several groups working on different kinds of microbiomes. So altogether, we publish a paper on the standard operator procedure for microbiome sampling and analysis. And we are finishing the setup of protocol for the safe storage of microbiome. And in several cases, we are also working on the propagation and reuse of this microbiome for different applications. As regard the services and the education, we are trying to mimic as much as possible what produced at European level. So we, first of all, we set up a stakeholder survey to try to identify the needs of the industrial Italian stakeholder. And then we set up an online catalog of the services and of the training course. And we have already launched the first Italian transnational access call. And as regard communication and dissemination, we have now our website, and we have a lot of activity for the dissemination of our result. So what the creation of a national node has been really very important for us. First of all, because indeed in January 2025, Italy decided to enter in the MIRRI community. So the ministry recognized the importance of our Italian network for our country. And this also was really very important because immediately we were recognized as a network, and so we were asked to collaborate with other ministries, for example, with some ministries regarding the biotechnological risk, or for the one working on food system for the creation of the catalog of the microbial resources useful for food production, and so on. And so we are working in synergies and collaboration between different policymakers at a national level. We are setting up a different working group within our network to exchange best practices and so on, and to work together to solve different problems. We are trying to harmonize and improve our communication activities towards the scientific community, but also towards the industrial communities, and also towards the lay society, because microbiology is, let me say, a difficult topic. Most of the persons are scared about microbes, and so we have to change their point of view, showing how microbes are powerful and positive for our planets. And this is something really important to making the lay society more positive towards different biotechnological application. And finally, we are working on the long-term sustainability of our network because culture collection are really wonderful, but are also very costly. And so we have to understand how to get money to continue to work all together. So here is, I try to summarize the positive the positive effect of our national network for the scientific operators. As I already told you, for the first time in Italy, there is a single access point where you can find all the Italian microbial resources and relative metadata. You can also find all the cutting-edge technology which are available for any kind of purpose. And of course, the staff of each of the microbial biobank is available for consultancies and other things. And you can also find the list of the training courses. For our system, so for the staff of a different cultural collection, we offer some tools to increase the management of the cultural collection, increase their visibility, and also their profitability. We created also some training courses which are really specific for the staff of the collection. And in general sense, for the MIRRI network and the European community, we can offer, the Italian network is really very skilled in different technique and application that can promote the industrial process through microorganism valorization. That is, so in some way, I really think that the Italian network is very strong and has also some peculiarities which are not present in the other countries in Europe in terms of microbial culture collection. And we try to evaluate the impact of the Italian national node in different ways. And as I told you, one of the most important things I want to underline is that most of the Italian strains preserved in the Italian microbial biobank are unique strains, so are not copies of strain already deposited in our collection. And many persons working in the Italian biobank are really keen in the valorization of the different group of microbes, so they can be really very useful for the design of new product and process to foster bioeconomy and green transition. And also, of course, in dialoguing with the policymaker to try to understand how it's better to face different societal challenges. And here I just want to give you some data about the industrial stakeholder survey. I just want to underline that more or less 600 industry replied to this survey, so a quite high number of industry. And here you can see the different sector this industry belong. And here are the type of services that they would like to get from us. So it was very important for us to try to understand which are the services we have to set up for them, and also the type of training. So now we are more aware of what we have to develop for the industrial sector to help them in facing their needs. And of course, we are also aware that we are just at the beginning of our pathway. We need to continuously improve the quality management system of our collection. Just to give you an example, when we start with the project, only 5 collections were already certified for their kind of services and activities. Right now, there are 4 or 5, I can't remember anymore, collection reach the ISO standard certification. And so this is a way to really, and we share everything among us. So this improvement of the quality management system have to continue in the next year, and we need to work much better with the industrial stakeholder, trying to engage much more this industrial community with our activity. We have to improve the fairness of our data, and of course, amend capacity building with cutting-edge technology. And of course, increase the number of training courses we can provide to both very specialized person or to a more general audience. And of course, also find money to continue to work all together. So first of all, I want to thank you and the organizer for this opportunity, but I also want to thank you, the wonderful Italian community of MIRRI, because I'm really proud to be their coordinator. Thank you.
Thank you so much, Cristina, for this presentation, very insightful. Are there any questions in the room about the possibility that MIRRI can offer you an Italian network? Especially from the bioindustrial partners, are there any questions? Come on.
Don't be shy.
There are a lot of possibilities of service, of access to services and TNA, and it's very nice opportunity to ask the coordinator. Yes.
There is one thing that
Come here, please. There, come on, then you can make some exercise, oxygenation.
There's one thing that I have been looking out, and it's tough to find that culture collection that assures that you have authentic culture when you buy it, and it's really assured or certificate that it's authentic. Do MIRRI have some culture collection that gives this service?
The certification of authentic
Yeah,
microorganisms.
that guarantees that's authentic. I've been looking for
Okay.
one culture like that, culture collection like that, and I haven't found it around Europe.
I'm a mycologist, and I can tell you that it's really very difficult to give you this kind of assessment because, you know, any fungal culture could be a small microbiome in the sense that the culture could be, let me say, pure in a general sense, but then inside fungi can always host some endobacteria and mycoviruses. So, which is your definition of arsenic? Because if arsenic is the definition of it's not contaminated by other fungi, it's okay. I can provide you this kind of confirmation. But if you're asking me if they could host endobacteria or viruses, I should do specific analysis on any strain, and this is not still now available. So if we are already aware that fungi host endobacteria or viruses, we already specify in the catalog, but otherwise, when we say that there is a fungal, the collection means that there is no other already identified microorganism in the same culture. I don't know if I replied to your question.
That's basically it. Like normally when we want to get an arsenic culture, we really want a culture that has just that one organism, not any endo organism or a symbiotic organism together. Just for algae and for
Yes. So if I may, so the culture collection in MIRRI EU network are a culture collection of very high level in standard qualities of procedures and strains provided. So of course, as Cristina said, it's not always the case, but you can have the highest level of quality offered by culture collection, no, Cristina, through MIRRI network. So you can even browse the catalog and see if the culture is arsenic or not. So the catalog of MIRRI, Eric, can offer you this possibility, and you can complain, of course, if it's not.
Yeah, that's the thing that on most platforms of culture collection, you don't have like the information if it's arsenic, completely arsenic or not. It's really tough information to find. That's why I was asking.
I'm not an expert on chytrid, but what I can add is that many chytrid are biotrophic organisms which need other organisms for be able to be grown in culture. And so that could be another issue in this case.
It is, of course.
Yeah. But you can browse the catalog for sure and see if you find this level of quality that you're looking for. Okay. Because you have a lot of information. We will talk later about the minimum data sets and that each microorganism must have for each to be displayed through MIRRI catalog. So you have a minimum data set of quality information that are provided through MIRRI ERIC platform.
Okay, thanks.
Yes, another question?
I have one point to the topic. The one point is to obtain.
Sorry, would you mind? Thank you, Luca. Another question is coming, and the person is actually approaching the microphone. Really.
Okay.
Okay. The one point is to obtain the arsenic culture, if it is indeed possible. But the second point is to be able to maintain for a long time the arsenic culture. So I think if you ask about the if we have ethnic cultures for different purposes. I think this information should be circularly updated. For example, every month contamination check should be conducted, and this information should be included in the database about this, because the one case is to obtain the ethnic culture, but what about if we are able to keep the ethnic culture for a long time? Thanks.
I think that's almost impossible.
I try to reply in this way. In principle, any strain that is published in the MIRRI database should be an ethnic culture. It's the first point. Then we are human beings, so we can make a mistake from time to time, but in principle that is, and if any collection deliver a contaminated strain, you can request other copies or so on. So there is no problem from this side. Usually you have to sign a material transfer agreement in which you state which is the reason you are searching for this strain, and if you are using it for research or commercial application, because in case of commercial application there could be some negotiation, and again you have to be careful about legal compliance, so for example Nagoya Protocol and so on. So when we deliver a strain, we ask you several things, including if you are you have the equipment to manage, for example, pathogenic strains and so on, because you have to accept the biological risk on the management of these microbial resources. And and last but not least, we provide you the strain, and in general, there could be also in the catalog some very easy field like medium for the growth of this organism or so on. But if you need the setup of specific protocol for the maintenance of the strain, that's it's up a collaboration with the different cultural collection.
If I may add, so the culture collection in MIRRI network has technician personnel and quality management standard for long-term preservation. So the collection in MIRRI, Eric, has the capability to keep the strain for long terms and to check for quality. If you want to build your biobank or culture collection, then if you want to achieve MIRRI standard, then it needs to be personnel that is able to keep for long term. But as Cristina said, there is really few stuff about long-term preservation and quality control on zoosporic parasites, on microalgae. So there is a lot of work to do, and this is the possibility to access with DNA, with the catalog of service, to develop this kind of collaboration with the cultures that has this standard quality and to enhance. So it should be a cooperative process to build up knowledge, no? And I can also provide the SOPs with the materials that we developed with MIRRI IT with all the participants. So you can find some of the SOPs that have been developed by MIRRI. I think we are running a little late, so thank you so much, Cristina, again for your presentation.
Thank you and bye. I have to fly to Portugal.
Yes, and spread the news.
Yes, spread the news.
Thanks a lot.
Keep up Italy in Portugal, okay? MIRRI,
Yeah, sure. Good luck for the next days of the Congress, of the workshop.
Thank you.
Thanks.
Bye.